Hey, so hello everyone. Welcome to A World Without MND webinar, our annual webinar. We're about 50% of the people who wanted to attend have now logged in, so we'll get cracking because we've got a very interesting program for you tonight. So let me just start showing the order of tonight. So hopefully you can see our first slide with my welcome for you all. We have two other speakers tonight. We have Kelly Williams, who's a very talented genetic scientist here in our MND Research Centre, who's done some extraordinary work, and she's going to talk about this incredibly powerful tool that we've developed over the last 10 years at Macquarie, which is our MND Biobank. And then it gives me great pleasure to introduce Stuart to you, who's done some spectacular work on using a very routine tool in every clinic and hospital, that of the MRI, and has developed some stunning techniques to help us use MRI analysis to pick apart motor neuron disease. And then this year we'll have a nice long Q&A session. So if you have questions, please submit them. Any questions, whether it's relevant to the topics of the three speakers tonight or other questions, please send your questions in. If we don't get to them tonight, then I will ensure that we get an answer to your question directly to you over the next couple of days. So tonight is being recorded, and Jackie will put into the post a link to our annual report that was released just in June this year, that outlines the depth and breadth of what we do here in the Motor Neuron Disease Research Centre. So in the chat, you'll see that there's a link, and if you want to have a look at our annual report, you can download that. And I encourage you to do that, to look at the administration of the centre, the finances, and the spectacular work of both the early, mid, career researchers that occurs here at Macquarie. So I'll start by talking about a clinical trials update. So here at Macquarie, over the last year, we've had a very intense trial program, and right at the get-go, I have to thank Richard Gann and Paula Ordonez Adantuaga, who are our two clinical trials coordinators here in neurology. And so they work tirelessly for us to be able to perform these clinical trials. And we do so in collaboration with the clinical trials unit here at Macquarie. So I'm going to touch on one of the trials in great detail, and then some of the other trials we'll just touch on briefly, and then where we're heading to next year. So firstly, I'll talk about the 3K3 APC study, which is an in-house designed and in-house funded and executed trial of the use of a novel therapy in motor neuron disease. I'll touch on the Atlas study, which is a pre-symptomatic trial of an antisense oligonucleotide, a therapy to help in motor neuron disease that is produced by a mutation in a gene termed SOD1. We now have a lot of patients on expanded access for tofersen who are already symptomatic with their motor neuron disease. I'll briefly talk about the WAVE study that's just been terminated, and now that's an antisense therapy for another inherited form of motor neuron disease called C9orf72. And then I'll also touch on the MEND study and the Lighthouse study. And we have another couple of studies that will be starting hopefully in Q1 that are targeting different aspects of motor neuron disease. So just to put it in context, in 2020, more people died in Australia from motor neuron disease than died from COVID-19. And in 2020, motor neuron disease killed 1 in 200 Australians. Now, we do not have an accurate number of how many people have motor neuron disease in Australia because at the moment, motor neuron disease is not a notifiable disease. And that's something that we're working on, hopefully, with the new state government. It's a little bit more common in men than women. It respects no age. Our youngest patient in the clinic was 19 and our oldest was 93 at symptom onset. Unfortunately, our lifetime risk in Australia of developing motor neuron disease is 1 in 200. So that means that you're more likely to get motor neuron disease than you are to win on a scratchy that you buy at the newsagent. The risk of motor neuron disease is increasing in Australia. So when I started working as a doctor in 1989, it was about 1 in 500 Australians died from motor neuron disease. And it's important to remember that whilst 10% of motor neuron disease is genetic, 90% is sporadic. And as our gene pool is relatively stable in Australia, that 250% increase of motor neuron disease has to be environmental. When we started here at Macquarie in 2010, we looked after 60 patients with motor neuron disease, and I thought that was busy. Currently, we now see about 100 new patients a year. And at the moment, we look after about 185 patients with motor neuron disease. We now look after 93 families with a genetic cause of motor neuron disease. There are no therapies that significantly alter the progression and the fatal nature of motor neuron disease, despite approvals from the FDA of drugs such as Radicava or edaravone. As I said, 90% of patients have sporadic motor neuron disease, although we now systematically look for previously unrecognized genetic causes of motor neuron disease in every patient that comes to the clinic. There are multiple mechanisms that are suggested to be involved in the cause of motor neuron disease, but unfortunately, we still have a dearth of therapeutics that dramatically slow and stop this disease. Over the last few years, we've developed and executed a trial of a drug called 3K3A APC in ALS. So this is a completely Macquarie designed and executed study. It's a collaboration between Macquarie and a biotech company called ZZ Biotech. And the funding of this has also solely been driven by Macquarie, and the funding of the trial was completely provided by the Far East Climb for MND. The concept of the trial is that inflammation is a central component of the pathogenesis of motor neuron disease. And this inflammation is pretty much determined by microglia, and they detect motor neuronal injury and also produce an effector response that damages motor neurons. Inflammatory microglia and monocytes have been considered as therapeutic targets, but really only a handful of trials exist where the agent tries to attenuate microglial activation. We can measure activated microglia in the central nervous system by looking at a protein that's expressed on the surface of microglia called TSPO. And we have access to a ligand called FEMPA that enables us to measure these activated microglia using a PET scan. Now, activated protein C floats around in all of our blood. It's a serine protease that has multiple actions, including anticoagulant, anti-inflammatory, and neuronal protective activities. 3K3A APC is a modified form of activated protein C that reduces its anticoagulant ability and retains its anti-inflammatory properties. And currently, this medicine is in trial in the United States as a salvage therapy post-stroke. Activated protein C binds to the endothelial surface, where it's translocated through the endothelium and produces anti-inflammatory effects mediated through the PAR receptors to reduce microglial activation. In addition, activated protein C repairs leaky blood-brain barrier, and it also improves neuronal survival. And there's been multiple preclinical studies that talk to this. This trial design was to work out whether this therapy is safe to give people with motor neuron disease. Now, to wind us back to 2021, when this study, and 2020, when this study was initiated, we were in the middle of COVID. And so the design of this study was to bring patients into hospital to give them an escalating dose of 3K3 APC and to try and make sure that this is safe and to see whether we had any ways of measuring whether 3K3 APC actually had a biological effect in motor neuron disease patients. We used several different methods to do that, including MRI, 18F FEMPA PET imaging. We've developed an in-house marker of monocyte activation, and I'll show you a little bit of that. And we also have measured multiple markers of inflammation, including cytokines, chemokines, kynurenine markers, and neurofilament. So the 16 patients who came into this hospital here had to come and stay with us for 10 days. So they had to see me every morning and every afternoon for 10 days. And this was a huge commitment of these 16 patients because it was in the middle of COVID and the hospital was quarantined. You couldn't come in, you couldn't come out, your family couldn't visit. And patients came in for screening. And then on day one, they had research bloods. On day two, they had an MRI scan and a PET scan. On day three, they had a lumbar puncture and we started treatment with 3K3 APC. And then the following week, we repeated all those assessments again. And this is a summary table of the 16 people who gave up a week and a half of their life to participate in this study. The Safety Monitoring Board concluded that there were no significant issues in these patients who received 3K3 APC. There were no major safety problems. Some of the patients had a bit of headache, but really there was no other safety concern regarding 3K3 APC. So the major goal of the study was to show that this medicine was safe and well tolerated in a small ALS population. So to come to the secondary outputs of this study to see whether we had efficacy. So this is a time activity curve of microglial activation in the motor cortex. And the blue line is pre-treatment and the post line is, and the orange line is post-treatment. And in the two PET scans between time points in each patient, we really couldn't demonstrate that there was any difference in the time activity curve as assessed by PET scanning. Now, although this is seemingly a disappointing study, what this study does tell us is that we have very accurate and reliable measurement of 18F-FEMPA activity in the motor cortex. So our reproducibility in this study was extremely high. We didn't really think that 2.5 days of treatment with 3K3 would change 18F-FEMPA uptake in the motor cortex, but this was the first time that this ligand was used in motor neuron disease to see whether there was any change in the motor cortex. I'm going to leave it to Stuart to talk more about the MRI parameters that were used in this study, because we particularly, Stuart and Tom Welton and the team represented here had developed a technique to identify very early changes in the motor cortex of patients with motor neuron disease. And we were particularly looking at this as well as changes in the blood-brain barrier to see whether 3K3 would produce any change. And Stuart will talk about this. It's really quite high tech, but Stuart will talk to this very elegantly in about half an hour. We've developed an in-house technique of looking at monocyte activation. So monocytes in your blood are interchangeable with the microglia in your brain. And the panel on the left shows the hyperspectral autofluorescence of monocytes of a patient prior to treatment with 3K3 and the panel on the right shows the spectral shift of patients after they've received 3K3 And we can measure this to demonstrate that hyperspectral fluorescence of monocytes shifts dramatically as a result of 3K3 But more importantly, because the second monocyte experiment was conducted 4 days after completing 3K3 therapy, this is an actual prolonged effect metabolically in monocytes. So the two top graphs are the data taken from all of the monocytes, so 5,000 monocytes from all subjects pre-drug and post-drug, and we have an accuracy of about 84% of working out whether those monocytes are from a person before therapy or after therapy, which is pretty good for a diagnostic test. But we went back and analysed the mean data derived from each patient because of the heterogeneity of motor neuron disease. Some patients have more active disease than others. And when we actually derive mean data, we have a 100% ability to discriminate whether patients had received therapy or not. We looked at 71 different cytokines and chemokines, such as interleukin-6 and interleukin-1 beta, but we were particularly interested in demonstrating a difference between doses in interleukin-17 and interleukin-23, and these two cytokines are implicated in the pathogenesis of motor neuron disease. So in the top line, you can see that interleukin-17 looks like it has a trend of going up. I'm not sure whether that's a real effect due to the small numbers in the study, but there was a clear difference between patients who received 30 milligrams of 3K3 And these results were echoed in the kynurenine tryptophan ratio, where, as you can see, pre-therapy versus during therapy versus post-therapy, our KT ratio is reduced, and this indicates a reduction of inflammation that's a prolonged effect as a result of 3K3 therapy. We looked at neurofilament. So neurofilament in visit 2, which is pre-therapy, visit 5, which is during therapy, and visit 8, which is post-therapy, didn't really change during the study. And neurofilament is a marker of the degree of damage of motor neuron disease. Now, when we look at individual patients, so the first 3 bars on the left are from patient 1, the next 3 bars from patient 2, the next 3 bars from patient 3, and so on and so forth, you can see that there's difference between patients. When we looked at and some patients had very high neurofilament levels, indicating a big degree of neuronal damage, and other patients, such as the patients down here, with very low levels of neurofilament in their blood. When we looked at neurofilament levels and looked at survival in the study, there was a very potent effect of neurofilament level in predicting survival. So where to from here? So the next study that we have on the whiteboard is to do a 24-week study of weekly intravenous 3K3 APC, because with a disease like ALS, treatment over 2.5 days is not going to change the disease. We need to treat patients for a prolonged period to see whether there's an effect on the rate of progression of disease. And we have now very accurate surrogate markers of disease activity that will enable us to try and pick that apart. We were unfortunately unsuccessful in our FIGHT MND application, and so we're trying to cobble together money to get this up and running. We estimate that the study will cost us around about $2 million to execute. We hope to start it in the beginning of next year. And so lastly, touching on the ATLAS study, which is a study of preclinical tofersen therapy in patients with mutations in SOD1. So we're now the third highest recruiting site in the world for this study. And it's a really tremendous study trying to get at reducing the destruction of motor neurons from mutant SOD1 before you have any weakness. The WAVE study, unfortunately, was ceased because of unexpected adverse reactions in some patients receiving a similar antisense therapy for a different gene, C9orf72. We're in the final furlong of the monopantal study. Monopantal is a therapy that aims at improving protein metabolism in cells through the mTOR pathway, and we await the study results with bated breath. We're currently in the middle of the Lighthouse 2 study, and you'll remember that the Lighthouse 1 study commenced in 2016, and this is now an international multi-centre study. Although Australia has the vast, or the majority of patients that are now recruited into the study, unfortunately, we're not even at 50% of recruitment, and our first patients will finish their initial 2 years in 2024. We hope to have 2 new agents into the clinic in the beginning of 2024, SPG 302, and you would have seen the press that unfortunately was trumpeted around the country last month, a bit prematurely. And there's another very interesting potassium channel modifier produced by a company called Curalis that we're looking to start in the beginning of the year. So that's where I'll finish the little potted summary of where we are in clinical trials. It is a bit dense. The 3K3 APC study, we have great hopes that this will help with the inflammatory process that occurs in motor neuron disease. And we hope that we'll be able to cobble together the funding so that we can start this therapy as soon as possible. So if you have any questions about trials, just pop them in the Q&A function. And at that point, I'm going to introduce Kelly Williams. I've had the privilege of knowing Kelly for over 10 years. She's an extremely talented scientist and has done some seminal work. She had a tremendous publication in 2019 in Nature Genetics, I think it was, Kelly, with the monozygotic twin study. And she has done some incredible work on inheritance by descent and unpicking the genetic causes of motor neuron disease. But in addition, she's been a guiding hand into the development of our MND biobank that we started in 2013. That was a very ambitious project that is now quite a huge entity. And at that stage, I'm going to pass over to Kelly to tell you a little bit about the biobank and its help in unpicking motor neuron disease. Thanks, Kelly.
Thank you for that lovely introduction, Professor Rowe. And I'm very grateful to be able to talk tonight about something that I'm really passionate about, which is the Macquarie University Neurodegenerative Diseases Biobank. And so a biobank is really effectively a organised collection of biological samples. And so in the case of our biobank at Macquarie, it's from MND patients and control participants. And so a biobank has these biological samples and their derivatives, which are, examples of DNA, RNA, plasma, and serum. And then these biospecimens are then coupled with sample matched clinical data and demographic data. And so in 2013, as Dom mentioned, when the different research teams came together to form the Macquarie University Centre for Motor Neuron Disease Research, in 2013, we really saw this as it was critical to establish some sort of MND sample repository to support and facilitate our research. And we saw it as such an incredible opportunity to integrate with Dom's clinic, so within Macquarie Neurology, to access patient biospecimens to do our research. And so what we actually do, like the actual working of a biobank, is that when MND patients or family members or control participants will come into the clinic, we effectively collect samples from them. So we recruit them into the biobank through informed consent. We collect data from them, so clinical data, lifestyle data, demographic data, and then we actually collect the biospecimens. So these tubes are for blood. We get a urine sample as well as hair collection. And so these collections are then processed from our amazing biobank team, and they end up becoming these derivatives, which are then stored as a barcoded sample. So all of the different types of biological collections that we take are then processed in a different way, and these aliquots or derivatives that we end up with are then stored at different temperatures. So in this picture here on the bottom right, these are RNA samples that are stored in a -80-degree freezer on site at Macquarie. And so because, as Dom mentioned, we established the biobank 10 years ago, it's now grown into a very, very large and comprehensive sample collection where we have more than 60,000 aliquots, so more than 60,000 of these little tubes of biospecimens. And probably the most unique, exciting thing about our biobank is the fact that we have longitudinal biospecimens throughout an MND patient's disease duration. So if we look at this image here, if we imagine, this line here is a scale from when a patient is first diagnosed throughout their disease duration. There's MRI and electrodiagnostic tests that's conducted before diagnosis. And so, we are hoping that we can add MRIs into our actual collections, but at this stage, they are collected before diagnosis in a patient. And then throughout disease progression at regular intervals, generally maybe 3 to 6 month periods, we have a full biobank collection of all of these biological samples, and then their resultant derivatives, as well as clinical and demographic collections. So all of this data that we have here is collected throughout disease progression of an MND patient. We also do the same for control participants or family members who are also willing to be part of the biobank. And so having a longitudinal collection, as I said, it's quite a unique aspect. So there are multiple biobanks that exist. However, longitudinal collections are not something that most biobanks do. And so here's just a figure that's been generated by my incredible postdoc, who this is a way that we can actually visualize the collections from participants in our biobank. So if we consider along this axis here, these are all de-identified study IDs for a subset of participants. And then along each line is the point of diagnosis, and then the individual collections throughout the disease progression. And we're also, so not only are we able to see, I guess, the diverse, the breadth of collections that we have across MND patients, but we can also include other data that we may have for them. So here we have genome sequencing data. So I'm a geneticist, and I run the genomics and bioinformatics research at Macquarie. So we generate genome sequencing data for a lot of the patients. So this gives us access to their entire genetic code. But we also, as I mentioned before, prior to diagnosis, the patients can have MRIs of brain, spinal cord, and the neurodiagnostic tests. So this, I guess, is a nice visual representation of the types of samples and collection points that we have in our biobank. And sorry. And so here's a summary of what we have currently in our biobank. So as I mentioned, we established the biobank 10 years ago with the vision that it would become an invaluable resource for our research centre. We have more than 1,000 participants. So this is across MND patients, control participants, and family relatives of patients. This is the largest biobank of its kind in Australia, and we should be very proud that we actually have formal accreditation and certification from New South Wales Health as a biobank. But one, I guess, a caveat of having such an outstanding, high quality resource is that it's very costly. So the biobank requires an annual cost. It has an annual cost of $500,000, and a small portion of this money comes from cost recovery from researchers using these samples, but the majority of these funds come from donations, so from philanthropic efforts. And so once we've collected the samples into the biobank and collected the samples, processed the samples into the biobank, perhaps we've generated secondary data from these samples. So that would include, a whole genome sequencing data. These samples are de-identified, and they go on to collaborative research. And that's the whole, I guess that was the whole idea of why we set up this biobank in the first place, was not only we could facilitate our own research at Macquarie University, but also encourage national and international collaborations through our data. So to ensure that these precious patient samples are used in the best scientific way they can be, we have a governance of our Macquarie Biobank. So we have the Macquarie Ethics Committee overseeing the biobank. Our custodian, Professor Ian Blair, he is also the director of the Macquarie University MND Research Centre. We have a new biobank manager, Dr. Judith Hebbs, who's come to us from Westmead. So Dr. Sarah Furlong, who is down here in this image, she has been our biobank manager, I think, for 7 or 8 years. She's recently moved to the clinical trials department at Macquarie, so which is great that we have good contact within the clinical trials unit now. So Judith manages the biobank, and these are the biobanking staff. So these are the people that actually allow us to have a biobank alongside Dom's clinic. And so we have these individuals here. They collecting the samples, processing the samples, databasing the samples, managing the samples. So there's a critical aspect of our biobank. We also have a committee. So this is a scientific committee where so I'm actually a member on the biobank committee now. We perform scientific reviews of every application from researchers who want to use samples from our biobank. So whether they're within Macquarie University or they're national, international collaborators working with someone at Macquarie, every research project needs to be written up into a research proposal. It's then critically reviewed by the biobank committee to make sure there's a lot of scientific merit to their study, statistical cohort design to ensure that it's a powerful study that will get us some real biological answers. And so here's an example of, I guess, a study design of so my PhD student who's just undertaken a large-scale RNA transcriptome study of sporadic MND patients. And so what I'd like to outline in this slide here in this figure, we have all of the individuals are plotted along the y-axis, and then their point, so the interval within the disease duration is plotted along the x-axis. So we have individuals that in this pink shaded area that we would consider early in their disease onset, and then individuals in the yellow shaded area late within their disease progression. And so not only having the samples, but all this sample matched demographic clinical data allows us to be able to design cohorts like this, and then ensure that every aspect of, every covariate variable is able to be statistically matched. This slide here, it's probably, I feel it as someone that's been involved with the biobank from the beginning, it's something that makes me really proud to be able to present this slide, to show all of the diverse research ideas that are using samples from the biobank. So we have more than 25 research projects that have been approved to use Macquarie biobank biospecimens. And you can see here, the diversity. So we've got plasma, serum, we've got DNA, we've got RNA, monocytes across all of these different research streams. So this one slide in itself, proves to us that it was worth it to set up the biobank 10 years ago. And so here we're able to use these samples to collaborate all over the world, because our international people, particularly MND researchers internationally, have taken notice of our biobank and realized what an amazing resource that this is to support research. So these collaborators here are individuals or research teams where we have actually sent biospecimens or their derivatives directly. But if we consider data derived from our biospecimens, say genome sequencing data, or de-identified genome sequencing data, we have collaborators all over the world accessing data derived from patients from our biobank. And so a bit closer to home is talking about how we're actually using these samples within our centre. So here is a diagram with the Macquarie University MND Research Centre, where we have 8 different and diverse research teams alongside the multidisciplinary clinic, so Dom's clinic. And what I guess the key thing to take away from this is that the patients, so MND patients, the biobank is at the core of our centre. So, it's at the centre of the centre. So we have the largest MND clinic in Australia as Dom's clinic. So we're able to develop and build the largest and most impressive biobank in all of Australia. So this is our research centre. We have a whole lot of diverse staff that work across the centre. And the reason that we came together as a research centre alongside the clinic was so collectively we could be co-located and answer some really important questions in MND. And now this next slide I'm going to take you through is mainly to highlight how biobank samples, so samples coming into the biobank and samples coming out of the biobank into all of the different research teams to support collective research. So if we start with a disease gene discovery that we made back in 2016 in Macquarie biobank samples. So we've identified these mutations. There were other different research teams that then used our finding of this gene mutation to determine the prevalence in broader biobank samples across Australia, also genetic counselling considerations for these mutations. And so then these were, I guess, genetic, so DNA, RNA data was given to these research teams to use. If we then come down to our functional biology teams, they were using so maybe plasma, serum for proteomics, fibroblasts, skin cells, to biologically try to understand how this gene defect was actually was biologically contributing to MND. And so what these functional biology studies collectively found using Macquarie biobank samples is that this gene mutation, it leads to a defect in neurons, which cause accumulation of a specific neurotoxic protein. So understanding that then led the researchers at Macquarie to develop this potential gene therapy that actually negates this protein accumulation. And so the university is now supporting this therapy through supporting it towards clinical trials through a Macquarie spin-out company called Celosia Therapeutics. So this, I guess, is just an impact of this biobank in the samples that are feeding out across all the multidisciplinary teams to basically come from a gene discovery to a potential therapy. Now, the last part of my talk, as a geneticist, I would like to outline some of the exciting large-scale genomic studies that we've done recently, that we've driven here at Macquarie using biobank samples. So we collaborate broadly across the world, and these biobank samples, as Dom mentioned, have been used in really high-profile research papers in Nature Genetics. And so but this one was not in a Nature Genetics paper, but I think it's a really important research paper that I'd like to highlight that was driven by one of the postdocs in our research team. So more than 1,000 genetic variants or genetic defects have been reported in motor neuron disease. So this is over the last 30 years since the discovery of the first gene to cause MND, which is SOD1 or SOD1. So more than 1,000 genetic variants across about 30 or so genes. If we look at the genetic sequence of SOD1, it's 154 amino acids long. And what we have seen in MND patients over the last 30 years is more than 197 genetic variants have been described across the entire gene. So this is a schematic way of looking at a genetic sequence here. So this is the gene along here, and these are all the different mutations that have been found in either familial cases of MND, so those inherited families, or sporadic MND cases. So this is just one gene. So in this study, what we wanted to look at was across all of the reported genetic variants, how many are actually in Australian patients with MND. So we performed a large literature review to generate a comprehensive catalogue of genetic variants, other associated biological features of these genetic variants. And then we had a look, we actually used genome sequencing data from over 600 Australian MND patients to see how many of these genetic variants were present and which ones were present. And so what we found in this study was more than 40 reported genetic variants were present in sporadic MND cases. We found that a third of sporadic cases, so of our 600, a third of them had at least one of these reported genetic variants, and more than 7% had two or more. And because we have this associated clinical data with clinical and demographic data with our biobank, we then were able to look to see that these 7% actually had an earlier age of disease onset. I think an outcome that I'm actually very proud of that came from this study. So this is, a biobank sample driven study in genomics. The catalogue that we generated as part of this study was used for the last major update of the ALS online database. So this is managed by our collaborators in King's College London, and they used all of our data to do the major update for this database, which is used by clinicians, genetic counsellors, and researchers all around the world. So this is, a direct outcome biobank work. And so last, I just want to finish on our future plans with what we're going to do with DNA samples from the biobank. And so as Dom had mentioned, that we do targeted genetic screening for every single patient that comes into the clinic. And we want to expand that to actually do whole genome sequencing. So when we can look not only at the genes that are normally tested, but we can look across the entire genome to find either important genetic causes of motor neuron disease or potentially something that may make someone eligible for a clinical trial. So we really see that this is going to enhance research genetics reports. And as you know, I mentioned at the start of my talk, the different biospecimen collections that we have for each patient We are currently working alongside machine learning and AI experts within our faculty at Macquarie to actually integrate this data towards better understanding of longitudinal progression of motor neuron disease. Okay, thank you. Thank you, Dom.
Thanks, Kelly. That's a tremendous exposition of the work that's been going on in the biobank, and it's a tremendous platform. You know, I think actually we're closer to 80,000 samples, which is quite a breathtaking curatorial program. And as Kelly mentioned, it has had support from Macquarie University as the engine room, but the vast majority of the biobank is supported by you guys, by philanthropic support, which is really remarkable. And again, thanks, Kelly. So I'm just going to turn over to Stuart Greaves. So Stuart and I have known each other for a very long time, even though he looks about 20 years younger than me. We actually go back quite a long way. And I had the opportunity to go to a talk that Stuart gave in about 2015 when he talked about some of his tremendous work in depression and defining drug-resistant depression using MR techniques. And we got to talking at the end of his presentation. We had a discussion previously about Parkinson's disease, but our discussion focused around the fact that really the best tool to measure upper motor neuron disease damage at the time is a tendon hammer that was invented, over 150 years ago. So I said to Stuart, is there any way that we can use his skill and ability and turn it into focusing on motor neuron disease? And we've done some tremendous collaborative work, and it's been a great privilege to work with him over the last 5 or so years. And so I'm going to turn it over to Stuart, and he'll talk to us about the advances in diagnostics and measuring the progression of motor neuron disease using MRI. Thanks, Stuart.
Great. Thanks, Dom. Hoping you can see my screen okay. It would help if I shared. Pardon me. There we go. So I'm going to talk today a bit about mostly about the technology that I've been working on from all my career, really, and how we've applied it to MND already, and how we hope to apply it. So what you're looking at there is a picture of my very talented postdoc Tom Welton's brain, and you can actually see his optic fibers on the right of your screen, and how they combine in the brain to his occipital cortex, the hairy sort of filaments on the left. And that's actually still to date the highest resolution brain image ever, and we acquired that at the Mayo Clinic. We actually did that on the back of a lot of experiments at the Macquarie radiology practice, MMI, where I've done the bulk of my imaging research for the last 8 years. So I'm a radiologist, originally a biochemist and a MRI physicist. Radiology is really great for detecting changes in the brain when they're really obvious. And obviously that's of great importance if you have a dissection, like the person in the middle on the bottom, or hemorrhage like the 2 people on the top left. But in the situation that people with motor neuron disease find themselves, or some of my other topics, such as concussion and depression, it's actually not so great. And more often than not, myself and others deliver somewhat frustrating reports to clinicians. NAD, no abnormality detected. And the reason for that is in motor neuron disease is quite simply conventional radiology is really showing you mostly anatomy. And the key changes in motor neuron disease happen in the neurons, and it just takes time for these changes to be reflected in the physical properties of the brain. You do see changes, but typically they're late phase, and basically we'd like to do better. So my really lifelong push and commitment is to doing that. And I think that's really important. So if we just think about what happens with brain imaging in Australia, it's actually about a third of all MRI scans are for the brain. And as I said, a lot of these are for conditions for which brain images give a good practical benefit immediately, things like brain tumours, multiple sclerosis, stroke. But really they represent the tip of the iceberg. So as Dom mentioned, motor neuron disease affects 1 in 200 Australians, lifetime risk, and is deadly. Average survival is 2.5 years post-diagnosis. Depression's at the opposite end of the spectrum. It's extremely common. About a third of people get depression. About a third of people get dementia, and about a third of the population has had concussion. And yet all these conditions, whether they're highly prevalent or less prevalent but deadly, mostly present with normal studies. So how do we fix that? So my main integrative project uses the same technology to probe a lot of different diseases. And at the bottom of the bottom line is we're trying to use the technology that's available but isn't currently deployed to image the stuff at the heart of the matter, mostly the neurons and their functional correlates. So what you're seeing here is a picture on the left of the base data that we use, and on the right, a very zoomed in picture, quite on the scanner at Macquarie, of neural fibers crossing within the brain. So we've been pushing very hard since about 2015 and have greatly improved our tech to the point where we're extremely confident with the right studies and the right application. We can find signatures that can allow us to diagnose diseases earlier, and therefore to allow clinical trialists like Dom to have the signal to be able to do their work and get better biomarkers to see and detect changes earlier. So here's some of the data that I originally connected with Dom about, and this is my work on depression. So conventionally, depression would be an invisible disorder from a structural perspective, and it's actually clinically defined by 4 or 5 essentially clinical parameters, things like, have you had trouble sleeping? And yet here we are, we can do an MRI of the brain and actually detect physical changes in the brain that are as obvious when we analyze them in the right way as a broken femur. What we try and do with all our work is very unusual, in fact, in psychiatry and actually in radiology, as we replicate. So on the left, you're seeing the test cohort. On the right, you're seeing a replication cohort showing the same changes. And this is the kind of style of work we like to do. Similarly, in concussion, I started originally noticing that the areas shown in the little image on the insert on the left in concussion were very similar to those seen in depression. And we've done a lot of work looking at that. And it's very real. And just like some of the stories in MND, we have people coming in through my imaging program. This young chap was a straight A student, got a concussion in a basketball game, and with a really sensitive technique, we're able to see tiny microhemorrhages in his frontal lobe. And he's gone through the same research protocol that the MND patients go through. And what we're trying to do is make that much more routine in terms of picking up these changes, quantifying them, and actually bringing them back into science, into the same sort of settings as the biobank. In fact, I'm very excited about integrating with the MND biobank with our data. Here's some other work we've done looking at iatrogenic injury. And similarly, this often goes unnoticed, often isn't measured. These little dots are tiny little strokes that have occurred during procedures to fix aortic valves. Here's some other work we've done on people with congenital heart diseases, not usually thought to involve the brain, but when you look and when you measure with a quantitative technique, you can see that there are indeed cognitive deficits, and there is indeed a structural change, and this is related to the many procedures these people have. Okay, so in terms of the work we've done with motor neuron disease, the key technique we've found is one called diffusion kurtosis imaging. I won't go too deep into the physics, but essentially what diffusion kurtosis imaging does is it detects cellular change at a very fundamental level. And an easy way to think of that is if we looked at the distribution of water diffusion inside a cell, we can see changes in that using this measurement. So if red is the normal distribution of kurtosis, we can see that change during a pathological event and detect that and reflect that quantitatively. These are the basic images we analyze. It is extremely involved. I won't go too deep into it. We have about 4 or 5 different methods, and the idea is we combine those to reflect the different pathological aspects of the multiphasic and multifactorial pathway that we know to happen during MND. So another one of those is arterial spin labeling. And this is a technique where we can look at arterial flow without contrast in the brain. We've also started using a technique which looks at leakiness in the blood-brain barrier. And I think that's one that Dom and I are extremely excited about and has been shown to have good sensitivity. Another technique is quantitative susceptibility mapping. And why that's interesting is that we know that when there's cellular damage, iron leaks into the cells. And if we use this particular technique, which is an evolution of a more common technique used in routine radiology, and we look at the motor cortex, we can actually detect changes in iron concentration. And this is proven to be very valuable. In fact, combined with diffusion kurtosis imaging, we were able to generate an early diagnostic predictor of MND status. It's early days, so we're pretty cautious about how aggressively we want to promote it, and it needs further testing. But early signs are pretty good, with an area under the curve of 0.86, which is quite high. Sensitivity, specificity, and accuracy all in the mid-80s. And that's generally classed as a, open quotes, good test, and something we think we can definitely improve on. So what are we looking at moving into the future? So the work we did with our Mayo colleagues really proved foundational. So if you look at the top image, this is a way of representing fiber orientation in the brain. A simple way of thinking about it is larger, more coloured fibers represent bigger directional change in the brain. But this is the best we can do with standard scanners. And the bottom one was the best we could do with a magnet called the Compact 3T scanner, which we had a hand in developing with GE and the Mayo guys. And I think this is the future. And you can see this is about 10 to 20 times more sensitive and accurate in terms of representing these fibers. More importantly, in this data, we realized that there was a software technique of acquisition that we could actually apply on routine clinical scanners, which is in some ways more exciting. Here's some other work, which is more about what we can do with the data. And I think this is actually really relevant to MND. So in this case, we focused on the hippocampus, which is more of a marker for depression, a brain region associated with depression and dementia. But what we did is we formed a connectome. The connectome is saying which regions of the brain is this person's hippocampus connected to. And the sundial sort of representation here on the left is one way to represent that. And the dot and spoke model on the left is another way. But the bottom line is the dots and the spokes are showing where the common fibers are. So you could imagine applying this to motor neuron disease and looking for small abnormalities in motor cortex or other brain region changes. Here's some other work, slightly more boring, but no less important, to do with understanding how accurate our different methods are. Kelly spoke before about, and Dom also spoke about carefully measuring the variation in different techniques. Well, we're equally interested in that. And the question we really asked is how well can we measure the fibers that connect region A to region B, or even A to B to C in the brain, because that's really ultimately what we want to measure and be able to detect when pathology is inhibiting those connections. And the answers we got are somewhat complicated, but they greatly informed our protocol that we currently use, which uses a method that's much more sensitive and resistant to error in this area. So this is the sort of outcome that we're looking for, and the sort of biomarker that I think would greatly help monitoring progression of MND and potentially acting as a screening tool. So in this one here, we asked the question, what functional connectivity, so resting state blood flow changes and structural fiber changes were related to memory performance and what were related to emotional performance. And we were able to get moderately predictive metrics in a normal population and then replicated that in the human connectome project. And that's the sort of work we're currently focusing on with the motor neuron disease cohort. Just to finish, here's some other work comparing different protocols and looking at another candidate marker called the default mode network. And this essentially measures how much the standard resting state of the brain is perturbed in conditions. And we improved the detection of this network strength by about 50 or 60%. So we're very excited about applying this moving ahead. So I might just finish there. I hope that's been interesting.
Thanks, Stuart. Yeah, that was a tremendous tour of the ability of the MRI to help us unpick the biology of this complex and heterogeneous disease. I'm going to throw open to questions and answers if people wish. So if you just, there's a little button to type in your questions, and we have a couple that are already up and running. So I'll start out with the first one. So Damien has asked a question about the Monopantal study, and the company that's behind Monopantal have actually released to market some information of the biomarkers, and particularly neurofilament. Look, it's important to put the context of biomarkers in the size of the sample that you're looking at, and the Monopantal study, of which we're one of 2 sites in Australia, is a very promising study. So Monopantal is a mTOR inhibitor that attempts to improve protein metabolism within injured cells, and we're cautiously optimistic, but the Monopantal studies are still very early, and neurofilament is a very interesting biomarker for us to follow, and which is why we now measure that in-house. It is, if ultimately we can demonstrate that we can attenuate neurofilament levels by reducing them with a therapeutic intervention, that would be really good. But as you could see from my data, the variance of individual patients of neurofilament levels is indicative of how severe the disease is. Although the average survival is 27 months, the survival ranges from 8 weeks to more than 30 years. And our neurofilament appears to be a surrogate marker of disease activity, and attenuating that neurofilament level, I think, is one of the surrogate markers of neuronal damage. It's important to say that, neurofilament is not specific to motor neuron disease. We find neurofilament elevated in, for example, traumatic head injury. If you, to put it in a more recent context, if you were admitted to intensive care with encephalopathy from COVID and your neurofilament was above a certain level, it was associated with an extremely poor outcome, because it's a sort of like a marker of the degree of neuronal damage, not necessarily just your motor neurons. So a long-winded answer to your question, Damien. There's a question from Jane for Kelly. So Kelly, is it possible that you can get samples from other MND clinical care centres across the country for your biobank?
Well, thank you, Jane, for asking that question. At this stage in our biobank, we do remote collections for familial MND. So if we have MND patients in the clinic that have a family history, and potentially there's family members over the country, we will do remote collections. But one of the key aspects of our biobank is the data collection that happens in the clinic. So the short answer to that is, at this stage, no. However, we're part of a Australian consortium called SALSA, which is the Sporadic ALS Australia Consortium, and they have the consortium together has set up the ability to get remote collections, so remote sample collections they're normally from the major MND clinic within each state, sent to the University of Queensland to be put into a similar repository to what our biobank is, but it's only single time points. And we, as Macquarie samples, at this stage we make up the majority of the samples that are in the SALSA sample collection. So my suggestion would probably be that at this stage we won't be doing remote collections across the country, but SALSA would likely be interested in it. So.
And I might just jump in there too. So one of the important things is clinical ascertainment of the disease that the person has, so that there are lookalike conditions in motor neuron disease, and sometimes it can take quite a long time to sort that out. So the other thing, so the clinical ascertainment, given that the patients are diagnosed principally in one site, means that the diagnostic certainty is pretty high. We also have other lookalike diseases that we collect, like multifocal motor neuropathy. And we also have other disease control populations, such as Parkinson's disease, that feed into the biobank. And so even though the biobank effort started out as a motor neuron disease effort, it's actually now called the Neurodegenerative Diseases Biobank, because ultimately it will expand out to enable us to discriminate and look at biological measures in multiple different diseases. So no more new questions coming through, but I wanted to throw one to Stuart, if I could, and that is, do you envisage that the quantitative methods of MR will become, because pretty much everyone with motor neuron disease has MRI scans in their workup of their brain and spinal cord. And, you know, quantitation even of spinal cord from the French group has been, in terms of volumetrics, they argue that, you know, spinal cord volume is a significant prognostic marker. Do you envisage that serial MR analysis will be a useful tool in looking after our patients with motor neuron disease?
Yeah, I mean, it can and should be. And I think one of the things that is frustrating to me as a radiologist is we, you know, I don't want to be negative. I think radiologists do a great job. But one of the things that you see a lot in areas like cardiology, for instance, they're all into their metrics. You know, they have, you know, these scores and clinical algorithms that can help triage people into effective treatments. And even just rolling back to what you're talking about, you know, obviously one of the roles currently for MRI is looking for other causes of the person's symptoms. And I could see immediately even simple metrics, like relatively simple metrics like spinal cord volume, you know, motor cortex volume, I think could be useful immediately for pushing people into other diagnostic pathways, or even just improving the pre-test probability for other tests, so that people like yourself, when you see someone in clinic, could look at some numbers which you're very familiar with and weigh them up in terms of your decision about immediate, you know, what the next steps are. But for that to happen, you know, the barrier is that imaging happens in a distributed manner. It needs to be much more standardized and protocolized. One of the, again, from the cardiovascular area, one of the scores that's had huge impact is really simple. Like, it's the calcium score in the coronary arteries. It couldn't be simpler. And yet it actually is more powerful than the Framingham risk score, which is this super famous, you know, score for predicting the risk of heart attack. And you can do a simple test of how much calcium is in someone's coronary arteries, and that can completely re-stratify how you treat someone. So yeah, absolutely. And biobanks are critical, you know, for supporting, you know, building the evidence to push for that change.
Brilliant. Thanks, Stuart. Look, we've had a comment from Job. Thank you for your great work and dedication. It means so much to us. Well, look, Job, it's a pleasure, but, you know, it's a good start, but we're not there yet. We haven't fixed it. So thank you. Question from Colleen. Hi, Colleen. Thank you for an amazing insight update tonight. As a patient that was enrolled in Lighthouse 1, I know how important your work is. My question relates to funding. Dom, you mentioned that the new study's cost to commence at the beginning of next year will be in the vicinity of $2 million. How close are we as of that target today? Well, just don't leave your wallet around me because I'm a terrible thief. Where have partitioned around about 300,000. So we're, around about 15% of the way to getting started. But, my approach to this, Colleen, is that we don't have to wait till we have the money in the bank. I'm quite happy to run this stuff on a promise. Don't tell the finance department in Macquarie that that's the case. Okay, so no one ever heard me say that. But if we have enough to get running, the blowtorch on all of us every single day is that this year in New South Wales, 250 people will die from motor neurone disease, and I'm on a bit of a trying to think of the right word, absolute campaign to make this disease a notifiable disease. So yesterday afternoon, I saw a woman from Davistown up on the Central Coast who's lived there for 21 years, and she has motor neuron disease that started. She has been a carer in the community for a long time. And she had troubles doing up buttons. And long story short, she came to see me yesterday and she has motor neuron disease. Today, I saw a fellow who also lives in Davistown that I've been looking after for 3 years. And he lives 100 metres from the patient that I saw yesterday. Now, we're in the middle of starting the genetic assessment of the patient I saw yesterday. I already know that the patient that I saw today has sporadic motor neuron disease. So what's the likelihood of two patients that live within 100 metres of each other for the last 20 years, and they didn't know each other and I didn't disclose their identities, that's just a random event? So we have to pick this apart with our sophisticated tools, with our biobank effort, with our imaging effort. We have to use the epidemiologic data. And, many of you will have heard me talk about the Riverina as a microcosm for what's happening in our environment. And we estimate that in the Riverina, motor neuron disease has a 4-fold increased incidence over the last 10 years. Now, we need to make this a notifiable disease, and we need to be systematic in how we measure it. We need geolocation for patients because it's undeniable that this is environmentally triggered. And the point of it being potentially environmentally triggered is that perhaps this disease is preventable if it is environmental, but also if we understand the environmental causes, that will shed light on the biology of the disease so that we can dramatically slow and stop it. So if we can slow the progression of motor neuron disease by 50%, theoretically, we can quadruple people's survival from an average of 2.5 years to 10 years. And as Jane has just commented on the Q&A, in the area in which she lived, there were 5 people with motor neuron disease in a very small area. Now, the issue is that we need to make sure that there's not genes involved, and there still may be an environment and genetic interaction, but that's the importance of the oligogenic inheritance of seemingly sporadic motor neuron disease. But even then, there may well be an environmental contribution. Hello, Jen. How are you? A comment from Jennifer Kutas. Andrew Fordham was my brother. He died 10 years ago this November. And she says she's terrified that these people in Davistown are from Allura Waters Retirement Village. They're not. But Emma, we need to pick this apart. We need a heat map of where motor neuron disease is, not necessarily that the triggers in the environment are ubiquitously going to cause motor neuron disease, because we know it's only 1 in 200. Kathy asks, is MND notifiable anywhere in the world? Well, in France, there's an MND surveillance unit that's centered in Paris. And so in France, when someone is diagnosed with motor neuron disease, the surveillance center in Paris is notifiable, is notified. So in Australia at the moment, if you get influenza or hepatitis B or hepatitis C or HIV or monkeypox or, God forbid, COVID, that's a notifiable disease. And in fact, in 2020, if you had COVID, we sent police to your door. If you have a cancer cut out of your skin or you're diagnosed with lung cancer or breast cancer, then that gets notified to the cancer registry. But if you get motor neuron disease that kills you on average in 27 months, the Department of Health is not interested. So I have a meeting in the next week with one of the senior advisors to Ryan Park, and we need to knock this off state by state because I don't think you can do this from a federal level. And the Department of Health isn't interested in making this a notifiable disease because it's actually going to turn a very bright light on one of the problems of motor neuron disease, which is access to timely and expert diagnosis and care. So how can we get this made notifiable? Well, we need to petition Ryan Park, and we actually need to continue to bang the drum. It's not acceptable that 250 people are going to die from this disease this year, and the Department of Health doesn't care. So coming back to my opening remarks, because we're sort of heading into the final furlong, unless anyone's got any burning questions. In 2020, as I said, 747 people in Australia died from motor neuron disease. 700 people died from COVID. And we threw nearly $500 billion at COVID. In that same year, 2020, 16 people died from silicosis in Australia, which is tragic. But silicosis is a notifiable disease. So we should have the same notification system for a diagnosis of motor neuron disease. I was down in Leeton a couple of weeks ago filming with one of our patients, Rocky, who is a citrus farmer. I've had 2 other citrus farmers on the shores of Lake Wyangan with motor neuron disease. I have patients in Wagga, in Albury, in Narrandera, all throughout the Murray irrigation area with motor neuron disease, that it's highly likely this is environmental. So we got to turn the heat up on the guys who count this and cut loose resources so that we can do better. Goodness gracious, I was on the soapbox for a little bit there.
Dom, I actually lived in Leeton for quite a bit of my youth, so I'm deeply interested. Have you got any thoughts about what the potential environmental factor is there?
Well, we think, Stuart, that it's associated with water. Now, we don't know necessarily which element it is. It may be a combination of factors because, you know, our fresh water has been destroyed over the last 50 years. So 50 years ago, you could scuba dive in the Darling River. And at the moment, according to New South Wales Water, there are 6 harmful algal blooms or BMAA blooms in the Darling River area. Look, just on a tangent to that, Phil Camden said that he's been giving to the biobank, but we haven't done any environmental stuff. Look, Phil, the environmental stuff is run by Prince Alfred, and Roger Pamphlet has had a project called ALS Quest. So, you know, we don't yet have an environmental unit attached to the biobank. It's one of the things that we need to do to try and pick apart this environmental issue, you know, a surveillance unit. And yes, Phil, indeed, you know, you also lived in the Hunter, and the Hunter is grossly overrepresented in motor neuron disease. Look, in the last 2 minutes, I'd just like to thank everyone for logging in to the webinar. We are going to have this recorded and people will be able to access it so that you can listen to my dulcet tones as well as the tremendous presentations by Kelly and Stuart. I'd just like to thank Jackie and Anne and Andrew, who've been in the engine room in the back, just getting this up and running. If you have further questions, send them in, download our annual report. And again, over the years, thank you so much for your support, because we can't actually do this work without your support. So thank you.